Hi. So, this is the second segment of the lecture on memories and holograms. In the previous segment of this lecture, we talked about memories and how you can understand them as attractors of a network, and we introduced the idea of a Hopfield network and how you can store memories using the Hebbian learning or Hebb's rule, and how you can retrieve them. And we also introduced the idea of an associative memory, right, that human memories are closer to an associative memory and are very different from index memory, which we see in a computer. But we never said anything about how these memories are actually stored in the brain or where they are stored in the brain, and that is the subject matter of this second segment. So, in this segment we will talk about hippocampus, which is a very important memory related structure in the brain, and how it stores memories for the short term. Now, memory, we use the term a bit loosely, but there are different kinds of memories, and there is a very broad taxonomy or classification of memories or long term memories into procedural memories and declarative memories. In procedural memories, these memories deal with motor memories, for example, cycling. I mean, that is something you cannot explain how you do it, you just know it, it is in your motor system, whereas the declarative memories are something that you know it and you can express it often in the form of language. And here there are two subcategories. There are the semantic memories. These memories deal with the general knowledge, and episodic memories, these are personal reflections or memories of events that you have experienced in the past. So, hippocampus is a very important subcortical structure in the brain, which plays a major role in storage and consolidation of memories. It is called the scratch pad of memories because this is where you store information or memories for a short term, after which they are passed on to long term stores in the cortex. So, any damaged hippocampus is known to impair our ability to store and retrieve the declarative memories. So, hippocampus looks like this. It is located inside the temporal lobe, and you can see that blue region which denotes hippocampus, and on the right you see the actual anatomical structure of hippocampus on the left and or in the middle, and on the right extreme you see this little sea creature called the seahorse, and the word hippocampus comes from the seahorse. Hippos means horse and campus means sea monster, because of this appearance of this structure to seahorse, and it is named hippocampus. So, our earliest knowledge of the functions of hippocampus came from the studies of a patient known as Henry Molaison. For a very long time this patient was referred to by his initials HM, but recently the patient passed away, and since then they began revealing the full name of this patient. So, HM had his medial temporal lobe removed as a treatment for his epileptic attacks. There was a time when in the case of intractable epilepsy, people used to remove temporal lobe because the source of epileptic seizures or the epileptic focus used to be generally located in the temporal lobe. Surgical removal of this region is generally considered a solution for the epileptic attacks. So, his temporal lobe was removed and medial temporal lobe was removed, and after that HM suffered from two kinds of amnesias. So, one of them is retrograde amnesia, which refers to inability to recall old memories. So, basically if in this picture you see the time at which a lesion or in this case surgery has occurred, and retrograde amnesia refers to memories of memories that happen from the time before the lesion or surgery had happened. So, this patient had retrograde amnesia up to a period before the surgery has happened, but memories from much long before the surgery, right, some of them were intact. Then HM also had anterograde amnesia, which refers to ability to store new memories, that is after the lesion he was not able to store memories. So, in the picture below it shows retrograde amnesia or anterograde amnesia, but the patient HM had both kinds of amnesia. So, basically let us look at the relationship between hippocampus and cortex. So, as you have seen in the picture, it is a subcortical structure, it is located deep inside the temporal lobe, and it is a subcortical structure. Now, how is it related to the cortex? So, basically to put it very simply, hippocampus receives extensive projections or convergent projections from the cortex, and it also sends divergent projections back to the cortex. So, if you expand that picture a little bit and put in some of the anatomical regions both on the cortical side and the hippocampal side, the picture looks a bit like this. So, in the neocortex you have three major regions, parietal, prefrontal and temporal, and all these three regions send feed forward and feedback connections to parahippocampal gyrus and perirhinal cortex, and these two regions in turn send projections to the entorhinal cortex, which is not shown in this picture. Then the entorhinal cortex, which is considered the gateway to hippocampus, right, then sends projections deeper into hippocampus and to various hippocampal fields. So, hippocampus is basically a system for association. So, it associates different features or different properties of an object. So, very often when you experience an object, when you receive the sensory information from an object, this information can be in multiple sensory modalities. So, for example, if you look at a ball, if you are looking at a ball that you are holding in your hand, right, your visual information tells you that the object is round in shape, it has some kind of a color, probably red, and it feels hard or it feels soft or it is warm or hot or whatever, right. So, you receive information from the object through multiple sensory modalities, and all these modalities you combine into a single percept of that object. So, for example, if you are holding a cricket ball, right, it is red in color and it is round in shape and it is hard, right, in terms of its touch. So, all these properties are combined to create this construct or a concept called the cricket ball. Okay, so, the sensory properties are processed in the cortex in various sensory areas of the cortex. So, for example, color is probably processed in the V4 region of the visual cortex, and shape is processed in the infratemporal area of the visual cortex, and hardness or the touch of it is probably processed in the primary and secondary somatosensory cortices S1 and S2, SS1 and SS2. Now, these primary sensory areas project to higher sensory association areas in the inferior parietal, and from there through these stages that we have just seen, right, these cortical stages, these areas project to the hippocampus. So, the sensory properties are represented in the cortex and they all converge to the hippocampus, and in the hippocampus you code for or represent a high level abstract representation of the object. Here that is a cricket ball. So, basically hippocampus receives a compact high level representations of the object from the cortex. It also receives reward related information from the prefrontal cortex and subcortical structures like amygdala and ventral tegmental area. So, we have seen in some of the previous lectures that the neurons of ventral tegmental area, which release a substance called dopamine, right, represent reward. So, these areas and also prefrontal cortex, they project to the hippocampus, and here the hippocampus is informed about the reward associations of the object. So, in hippocampus has access to the reward information and also access to the sensory information from the cortex. It combines these two forms of information and decides if the information is worth storing for longer term back in the cortex or must be discarded, because one of the things that decides whether you store information or not is some kind of a significance or salience of the information. So, if otherwise as we say, for example, go down the street every day, you meet so many people or you see so many things, and all of that may not form part of your long term memories. So, unless you find something interesting or salient or significant, right, in some sense, right, your brain does not decide to pass it on for memory storage. So, like I said, the large parts of the neocortex project to parahippocampal gyrus and perirhinal cortex, and these two cortical areas in turn project to entorhinal cortex, EC, and EC is considered the gateway to the hippocampus. So, you can see that in this picture, you can see the two cortical stages, and then the input to that goes to entorhinal cortex, and within the hippocampus, right, you see a loop like architecture. So, from entorhinal cortex you project to the dentate gyrus, or which is shown here in this picture as dentate granule cells. From there cells project to CA3, and from CA3 they project to CA1, and CA1 they project to subiculum, and subiculum projects back to entorhinal cortex. So, it forms a loop of connections. There are more connections within hippocampus, and that we will see in a moment. So, like I said, entorhinal cortex is the gateway to the hippocampus, and from there you have a sequence of projections from EC to dentate gyrus to CA3 to CA1 and subiculum before returning to EC. Now, EC projects back to the neocortical targets via the parahippocampal and perirhinal. So, you can see that hippocampus receives compressed representation of the information arising from the sensory modalities and combines it with the reward information and sends the information back to neocortex for long term stores. So, if you look at some of the pathways within hippocampus, we just a moment ago we described the connections within hippocampus as a simple loop from entorhinal cortex going via all the hippocampal fields and returning back to entorhinal cortex, but actually there are more connections than that. See, for example, the entorhinal cortex projecting directly to dentate gyrus, this projection is named a perforant pathway. Then the dentate gyrus projecting to CA3, right, this projection is called the mossy fiber pathway. Then CA3 to CA1, this projection is called the Schaefer collaterals, and there is also a direct connection from entorhinal cortex to CA1, this is called the temporoammonis pathway, and from CA1 to subiculum and subiculum back to entorhinal cortex. So, you can see that within hippocampus there are several parallel fibers, and so EC also projects directly to dentate gyrus via the perforant pathway, and mossy fibers project from dentate gyrus to CA3, and from CA3 to CA1 you have Schaefer collaterals. Okay, so there are all these hippocampal fields, right, or subregions within hippocampus. Among them, CA3 region has been given a lot of importance and attention, and that is because in terms of the microscopic architecture of connectivity within the CA3, it has some distinguishing features compared to other hippocampal fields, right, and CA3 has very rich internal connections, or what are called recurrent connections, and this property of CA3 had drawn a lot of attention, and therefore, CA3 had been compared to the Hopfield network, because we have seen in the previous segment of this lecture that what is special about Hopfield network is its rich recurrent connections. So, every neuron in the Hopfield network is connected to every other neuron, and it is this kind of rich recurrent connections that enable this network to play the role of an associative memory. So, similarly in CA3 you have very rich connections among the pyramidal neurons of CA3. So, therefore, people have tried to draw analogies between the Hopfield network and its memory storage property and the CA3 and its probable contribution to the memory storage properties of the hippocampus. So, CA3 neurons have rich extensive recurrent connections, and for example, if you take the rat hippocampus, in rat hippocampus in the CA3 field there are about 300,000 pyramidal neurons, and each of them receives input from about 12,000 other CA3 pyramidal neurons. Whereas, if you look at the inputs from the EC to CA3, each of the CA3 pyramidal neurons receives inputs only from about 4,000 EC sources, whereas each of the CA3 pyramidal neurons receives input from about 12,000 other CA3 pyramidal neurons. That means, there is a very rich, so number of connections within the CA3 is lot more than the number of connections from outside CA3, that is from EC to CA3. So, each CA3 neuron, the pyramidal neuron receives input from about 4% of all the CA3 pyramidals. So, that is a very high level of recurrence compared to any other brain region. So, in Hopfield network model, since it is only a mathematical model, an abstract model, we have used 100% recurrence. That means, every neuron is receiving input from every other neuron, but that kind of a high recurrence is at least to our knowledge does not exist in the real brain. But even 4% recurrence is very high compared to any other part of the brain, and you see that kind of that level of recurrence in the CA3. And also these recurrent connections are subsequently found to be modifiable by LTP. So, in the last segment of this lecture, we have seen how LTP which stands for long term potentiation and LTD which stands for long term depression, right. These two mechanisms give you one way of activity dependent plasticity or activity dependent modification of synaptic strengths, and these kinds of synapses are found in many regions, particularly hippocampus. In fact, a lot of the early work on LTP and LTT was done on hippocampal synapses. So, therefore, considering these features of hippocampus and particularly of CA3, people have considered the hypothesis of CA3 playing the role of some kind of a Hopfield network in which patterns can be stored as attractors. So, now if patterns have to be stored in the CA3, then the network should be and for it to be useful as a memory, it must be operable in two modes, storage and retrieval, because so you need to first figure out how to store information in it, and then if you just store in it and you cannot retrieve it, then it's no use. So, you need to be also able to retrieve information from this network. So, how do you do both these operations on CA3 if it is acting like a recurrent network or a Hopfield network? So, to explain that, let us use a small analogy, what we call the classroom analogy. So, in a classroom you can imagine that the classroom operates in two kinds of modes. First one we call the learning the lesson stage, and second one we call it the discussing and reproducing the lesson stage. So, in learning the lesson stage, the teacher gives out her lesson and the students listen to the teacher and receive all that and and learn the lesson. So, during this stage the teacher's instruction dominates. So, only person who is talking during this stage is the teacher, and then all the students sit quietly and listen to the teacher, and there is not much chatter among the children, because the chatter among themselves that will distract them from receiving what is they are receiving from the teacher. So, children are in the state of receptivity and eager to learn the lesson. So, they are kind of open, and then kind of the brains are in a position to receive it and then store it inside them. So, this is the first stage or first mode, the learning the lesson stage or learning the lesson mode. So, in the second mode, so the children have learned the lesson and now they are asked by teacher to reproduce and express what they have learned in the previous stage. So, we call this the discussing and reproducing the lesson stage. So, in this stage the teacher does not say much, the teacher's instruction is low and the conversation among the children are high, because in this stage you would imagine that since they are very small children, a single child does not know everything that the teacher has taught. So, they have to talk among themselves, discuss among themselves, share whatever knowledge they have gained, and then as a group they express what they have learned. So, in this stage the conversations among the teachers are high and the children do not learn anything new, because now they are only recalling what they have already learned, they do not learn anything new. So, the brains are not in a stage of kind of plasticity, or they are not plastic in this current mode. So, so we have two modes here. One is storage mode, where teacher the children are all receiving information and not talking much among themselves, and their brains are in a plastic state, so that they can register or store what they have learned. And then in the second stage they are not receiving much from the teacher, but they are receiving more, that interacting more with other children, and the brains are not in a plastic stage and they are not learning anything new, they are only reproducing what they have learned earlier. So, these are the two modes in which this little group or this class can operate. Okay, so, if you take this analogy and apply it to the operation of CA3 neurons, and what does it look like? So, during storage the external input, so because CA3 is receiving information from higher parts of the brain, for example, the higher cortical sensory cortical areas project all the way to the EC or internal cortex, and then which projects to CA3. Okay, so, this is external input, and that during storage the influence of the external input on CA3 neurons is strong, and during this stage the influence of the recurrent connection should be weak, because a given neuron should be influenced more by the information coming from outside, right, and not be influenced too much by what the other neurons are saying to it, because the current connection strengths which the neurons have, right, represent the old information, and then that should not kind of distract the neurons from receiving the new information which is coming from outside. Okay, so that means the external input should be strong and more influential, and the recurrent connections among the pyramidal neurons of CA3, they should be weak. The third thing that you require in this for storage to occur efficiently is that the recurrent connection, that is the connections among the pyramidals of CA3, must be plastic. They are in a change, must be in a state of plasticity. So, they should be more easily modifiable by the ongoing activity within CA3. So, in the figure shown below, you can see that the external input coming from the EC, and that is represented by thick arrow. So, therefore, that is very influential. It is able to imprint itself onto the pyramidal neurons of CA3, and then the thin arrow which cuts across CA3, it represents modifiability or adaptation of CA3, that basically denotes that the recurrent connections are plastic, and then the loop kind of the arrow that bends backwards and returns back to CA3, it represents that the CA3 neurons have a very strong recurrent connection. So, this looping arrow represents the fact that there are recurrent connections within CA3. Now, for retrieval to occur, you need to have kind of opposite conditions to be met. So, during retrieval the external input should be weak. So, you see that the external input from EC to CA3, right, it is represented by a thin arrow in this case. That means, external input in this case should not be very influential, it should not influence the CA3 activity too much, and in this case the loop which represents the recurrent connections among CA3 neurons, this recurrent connection should be much more influential. So, recurrent connections are strong in this case, and third thing, recurrent connections are strong, but not plastic, they are not modifiable, but they are just strong. So, this is the kind of these are the kinds of conditions that must be met for retrieval to take. Here it is storage, and here it is all retrieval. So, now CA3 has two external inputs from EC via perforant pathway, and the other one is from EC via dentate gyrus by mossy fibers. Now, perforant pathway inputs are weaker than mossy fibers. Now, if you come to plasticity of CA3 recurrent connections, so acetylcholine, a chemical which is also a neuromodulator, is capable of controlling the strength and plasticity of recurrent connections in CA3. So, basically what we need is some way of changing the strength of external input, making it stronger, making it weak, some way of changing the strength of the recurrent connections, making it stronger, making it weak, and some way of controlling the plasticity of the recurrent connections. Okay, so, we see that all there are mechanisms for achieving each of them in place within CA3. So, we just said that acetylcholine is a neuromodulator. Now, mathematically neuromodulator is also a chemical, it is similar to neurotransmitter, but it is different in an important sense. A neurotransmitter basically is a messenger that carries a signal from the presynaptic side to the postsynaptic side, and the amount of neurotransmitter release or number of receptors on the postsynaptic side, all these together define some kind of an abstract quantity called the synaptic strength. Now, what a neuromodulator does, a neuromodulator actually is released onto a synapse, right, where you have a existing neurotransmitter like glutamate or GABA, a fast acting neurotransmitter. Now, the release of neuromodulator near a synapse onto a synapse modulates the strength of the synapse and can make it transiently stronger or weaker. So, and anatomically speaking, neuromodulator substances, there are four important neuromodulators. These are norepinephrine, dopamine, serotonin, and acetylcholine. And the memory research, the fourth one, which is acetylcholine, generally plays a much more prominent role. So, although all four do contribute to memory operations, acetylcholine is generally discussed more in the context of memory research. And these substances are released by small clusters of neurons located generally in the midbrain and some subcortical areas. And these clusters also project to, although these clusters are small, each having probably a few 100,000 neurons, right. These neurons project to widely distributed targets both in the cortex and in subcortical structures. Okay, so, for example, and like I said, ACH is a neuromodulator, and blockage of ACH transmission to hippocampus, right, is known to interfere with memory storage. So, ACH is it is released into hippocampus, and the ACH neurons project both hippocampus, that's their subcortical target, and they also project to the cortex. Okay, and so blockage of ACH is known to interfere with the memory storage in hippocampus, and also application of a drug like scopolamine, which is an ACH antagonist, that means it blocks the action of acetylcholine, is known to produce or induce a temporary amnesia. So, it will because it blocks ACH, it also blocks plasticity of the connections within hippocampus. It induces temporary amnesia or memory loss. So, scopolamine was administered in a memory task, and they found very interesting results. So, in this task, a bunch of volunteers were given a dose of scopolamine and were asked to memorize words, and some volunteers were given just a placebo, and so the two groups were given a bunch of words and to memorize. The group that was given scopolamine had little or no memory of the information presented during the test, and some volunteers even forgot about the trial. So, the memory loss induced was so profound that they even forgot that they had a test at that time. And scopolamine, when scopolamine was given before the experiment, right. So, the control subjects remembered about 45 out of 128 words, whereas scopolamine subjects, because of the action of scopolamine, its antagonist action of scopolamine on acetylcholine, these subjects were able to memorize only 6 out of 128 words. Okay, but when the scopolamine was given after the words were presented, that means the subjects are already learned the words and then they have taken scopolamine, right, then there is no impairment in performance. So, only when it is given before the task, right, it was able to influence negatively the performance. So, there is also evidence from electrophysiology that ACH controls LTP in the recurrent connections of CA3. So, seeing all these experimental evidence, Michael Hasselmo and co-workers have suggested that ACH satisfies the criteria necessary to switch between storage and retrieval, and that is thought to work out as follows. So, at high levels of ACH or acetylcholine, the input is strengthened, the recurrent connections of CA3 are weakened, and there is increased plasticity in recurrent connections of CA3. And at low levels of ACH, the input is weakened, the recurrent connections of CA3 are strengthened, and there is decreased plasticity in recurrent connections of CA3. So, that means at high levels of ACH, the CA3 is operating in the storage mode, because it matches all the conditions that we have asked for, right, for storage to occur. And at low levels of dopamine of acetylcholine, right, we achieve the conditions that are necessary for retrieval mode. Okay, so how do you say that? How do you say that at high levels of ACH, the input is strengthened and recurrent connections are weakened, and at low levels of ACH, input is weakened and recurrent connections of CA3 are strengthened? Now, how do we say all this? Okay, so it turns out that ACH has different actions on different parts of CA3. So, now so within CA3, if you look at the microscopic connectivity of neurons within the CA3, right, input to the CA3 pyramidal neurons from different sources are anatomically segregated in CA3. So, for example, a given CA3 pyramidal neuron receives inputs from other CA3 pyramidal neurons. These are the recurrent connections within CA3, and it also receives input from the EC. These are the external inputs to the pyramidal neurons of CA3. So, now inputs from EC to CA3 pyramidal neurons are located in a CA3 layer. So, there are different layers within CA3, and the external inputs to CA3 are located, these synapses are located in the CA3 layer called the stratum lacunosum moleculaire. Then the recurrent inputs from other CA3 pyramidal neurons, these are located in a different layer, different CA3 layer. These are known as the stratum radiatum. So, if you look at this picture, you can see how these different connections are segregated. So, you see at the top the external inputs from EC making connections with the dendrites of CA3 pyramidal neurons, and these synapses are located inside the layer stratum lacunosum moleculaire, and whereas the intrinsic inputs, that is the inputs of the pyramidal neurons among themselves, right, you see them in this middle layer, right, forming synapses among themselves, and that layer is called the stratum radiatum. And then the cell bodies of the pyramidal neurons are located in a third layer, which is called the stratum pyramidale. Now, it turns out that when you increase acetylcholine levels in CA3, it suppresses the synapses in stratum radiatum much more than it does to synapses in stratum lacunosum moleculaire. So, that means it suppresses synapses of the recurrent connections a lot more than it suppresses the synapses of the external inputs. Okay, so and also high ACH also increases LTP in CA3 recurrent connections. So, basically that means that it is at high ACH you are able to achieve the conditions necessary for memory storage in CA3. Now, okay, so we said that ACH is a neuromodulator, and increased ACH can to hippocampal complex or hippocampus, right, can increase memory storage in CA3. But what decides or what controls ACH release to the hippocampus or to CA3? So, ACH is released by two neuronal clusters, because we said there are these neuromodulators are released by small clusters of neurons located in the midbrain and other subcortical areas. So, ACH particularly is released by two neuronal clusters located in the basal forebrain. These are nucleus basalis, which projects to widespread cortical targets. It is shown in this simple cartoon figure here, and another cluster is medial septum, which projects to hippocampus. So, this loop between, so actually the medial septum projects to hippocampus, and hippocampus projects back to the medial septum, and that is called the septohippocampal loop. And when the branch from hippocampus to medial septum is activated, right, it is it suppresses neural activity in medial septum. Okay, and therefore, the loop from medial septum to hippocampus and backwards act as a self regulating system for hippocampus. And so, it is known that there is a feedback from CA1 field of hippocampus back to medial septum. So, you see that medial septum projects to CA3 and CA3 projects to CA1 and CA1 sends feedback to medial septum, and the effect of CA1 on medial septum is inhibitory. So, Esselmo and colleagues have proposed that the above loop is capable of making sure that ACH is released just when it is required. And how does it work? Basically, the idea is so CA1 is thought to be playing in the role of a comparator. So, we have been arguing all along in this segment that CA3 is like some kind of a hop field network, and it stores memories in the recurrent connections of CA3. And so, what the CA1 receives from CA3 is the representation of the stored memory within CA3, whereas CA1 also receives direct input from the internal cortex. So, this is the temporo-ammonis pathway, right, and that is the direct representation of the same pattern. So, CA1 is thought to compare the direct copy of the pattern from internal cortex and the reconstructed pattern, right, of the stored version in CA3. Now, the hypothesis is that if the original from the EC and the reconstruction from CA3 match, right, then CA1 makes this comparison and inhibits medial septum, and that suppresses or blocks release of ACH from medial septum. Whereas, if the original reconstruction do not match, so the original from the EC and the reconstruction from CA3 if they do not match, then CA1 sends feedback to medial septum, which activates medial septum, and therefore, that causes release of ACH. That release is released into CA3, as you can see in this picture. So, basically CA1 says, okay, the two patterns are not matching, and therefore, medial septum releases ACH to CA3, and therefore, that induces plasticity in CA3, and therefore, the pattern is stored in CA3. Okay, so that kind of completes the story. What this story says is, how or under what conditions, by what cellular and neurochemical mechanisms memory is stored in hippocampus. So, basically what it says is the key ideas of this theory is that the CA3 is functioning like some kind of a hop field network, and it operates in two modes, storage and retrieval, and ACH plays the role of a neuromodulator which switches the CA3 region between two modes of storage and retrieval, and the septohippocampal network or the loop plays the role of some kind of a regulator. And what this loop does is, if the stored pattern in CA3 and a direct copy from EC, if these two patterns match, then there is no release of ACH into CA3 because the pattern is already stored. But when these two patterns do not match, that means the pattern that is receiving is a new one or something that is not yet memorized. So, therefore, that causes release of ACH to CA3, and therefore, CA3 then stores the pattern in itself. But it is a very interesting theory, and actually I have got most of the material that I discuss in this segment from this reference. It is a book called Gateway to Memory by Gluck and Myers. But thing is, this theory has certain shortcomings. It does not address the question of saliency as a criterion for storage, because in the early part of this segment we mentioned that one of the key features, key determining criteria for storage of memory is the reward or saliency associated with the memory, right. Only when we find that the pattern is important for us, right, we store the pattern, and that kind of a importance as or saliency is very often represented by dopamine signals which represents reward. So, like I said, VTA has projections to hippocampus, but this current theory does not address exactly how that works. Okay, and also it is although it is an interesting theory, and but it is not fully tested, it is not fully realized in detailed computation models, so that you can really verify all these ideas in a detailed quantitative model. Also, this theory does not tell you how, it only tells you how information is stored in the hippocampus, but it does not tell you in what form and exactly how this information is passed on to the long term stores in the cortex. Okay, in this regard, I just want to mention that in my lab we have been working on an alternative theory of hippocampus and memory storage in hippocampus. So where we explain what is stored in the hippocampus and what exactly is passed on to the long term cortex and by what mechanisms. And our theory is based on reinforcement learning, so which is a very important form of learning, and I mentioned it before. The learning that happens in basal ganglia or is driven by basal ganglia can be explained very effectively by reinforcement learning or concepts on reinforcement learning. So, on the similar lines, we are trying to develop a theory of hippocampal function, where the memory storage and retrieval or memory consolidation or transfer of memory from hippocampus to long term cortical stores, all these processes can be explained very effectively by using ideas from reinforcement learning. But unfortunately, even our theory is right now at a preliminary stage, and hopefully by next year we are hoping that this theory will be well worked out and will be published. Thanks a lot.